The year is 1996. A lamb named Dolly is born, not conceived in the usual way, but created from a single cell of an adult sheep. This wasn't just a scientific marvel, it was a moment that ripped a page out of science fiction and pasted it into our reality. Dolly's birth ignited a global firestorm of debate, hope, and fear. What did this mean for humanity? Where were we headed? Now, decades later, we stand on the cusp of 2030, and the world of cloning is evolving at a pace that even Dolly's creators might find staggering. What does the future hold? Join us as we journey into the near future to explore what cloning might look like in 2030. To understand where we're going, we first need to appreciate where we are. Today, cloning isn't just about making copies of animals. It has branched into two main fields: reproductive cloning, which aims to create a genetically identical organism, and therapeutic cloning, which creates embryonic stem cells to generate tissues and organs. While reproductive cloning in humans remains banned nearly everywhere due to profound ethical concerns, therapeutic cloning is a hotbed of groundbreaking research. Scientists are no longer just dreaming of growing replacement organs. They are actively doing it in labs. Using a patient's own cells, they can create heart tissue that beats in a petri dish, or skin grafts that won't be rejected. This process, known as somatic cell nuclear transfer, the same technique used for Dolly, is now being refined to fight diseases like Parkinson's, diabetes, and severe burns. This is the foundation upon which the future is being built as we look toward 2030. One of the most exciting frontiers is the potential revival of extinct species, a process called de-extinction. Imagine seeing a woolly mammoth, not as a fossil, but as a living, breathing giant roaming the Siberian tundra once again. It sounds fantastical, but scientists are making real progress. Researchers at organizations like Colossal Biosciences are using CRISPR gene editing technology, a kind of molecular scissors, to edit the DNA of an Asian elephant, the mammoth's closest living relative. They are meticulously inserting mammoth genes for traits like shaggy hair, a thick layer of fat, and smaller ears, all adaptations for surviving the cold. By 2030, we won't have a perfect mammoth clone, but we could very well see the birth of a mammoth-elephant hybrid embryo, a critical stepping stone. The goal isn't just to create a biological curiosity. It's about restoring ecosystems. Mammoths were keystone species whose grazing habits helped maintain the grasslands of the north. Reintroducing them could help combat permafrost thaw and, in a small way, fight climate change. Beyond the mammoth, projects are also underway for species like the passenger pigeon and the thylacine, or Tasmanian tiger. The science is incredibly complex. It involves piecing together fragmented ancient DNA, overcoming the challenges of interspecies surrogacy, and ensuring these resurrected animals can thrive in a modern world. By 2030, the ethical debates will be just as intense as the scientific challenges. Do we have the right to bring these species back? What are the unintended consequences for existing ecosystems? These questions will be at the forefront of conservation biology. Now, let's turn to human health. By 2030, therapeutic cloning will have moved far beyond the lab and into clinical applications. The biggest hurdle for organ transplants today is rejection. Our immune system is designed to attack anything foreign. But what if the new organ wasn't foreign at all? Therapeutic cloning makes this possible. By 2030, we can expect to see personalized organoids, miniature, lab-grown versions of our organs become a standard part of medicine. A doctor could take a small skin sample from a patient with liver disease, use those cells to grow a mini liver, and then test various drugs on that organoid to see which one works best without ever putting the patient at risk. This is precision medicine at its most powerful. But it gets even more revolutionary. The ultimate goal is to grow full-size transplantable organs. While we likely won't be 3D printing complex organs like hearts and lungs on demand by 2030, we will see significant breakthroughs in bioscaffolding. Scientists are already using materials from plants, like spinach leaves, stripped of their plant cells, to create a vascular framework for human heart tissue. By 2030, we'll have advanced biocompatible scaffolds that can be seeded with a patient's own stem cells, coaxed into forming functional, simple organs like bladders or perhaps even patches of complex organs to repair damage. This could mean an end to transplant waiting lists for hundreds of thousands of people. It's a future where your own body provides the blueprint for its own repair. Agriculture and conservation will also be transformed by cloning technology. For endangered species, cloning offers a lifeline. When a species is on the brink of extinction, with only a few individuals left, its genetic diversity plummets, making it vulnerable to disease. Cloning can help. In 2020, scientists successfully cloned a black-footed ferret named Elizabeth Ann from the frozen cells of an individual that died over 30 years ago. This introduced much-needed genetic diversity back into the population. By 2030, we can expect to see frozen zoos, vast cryobanks of genetic material from thousands of species become a primary tool for conservationists. When a species numbers dwindle, we can use cloning to bring back genetic lines that were thought to be lost forever, giving these animals a fighting chance at survival. In farming, cloning is already used to replicate elite livestock, the prize-winning bull with the best meat, or the cow that produces the most milk. By 2030, this technology will become more accessible, and combined with gene editing, farmers won't just be cloning their best animals, they'll be creating designer livestock. Imagine cattle that are genetically resistant to diseases like mad cow disease, or pigs that are more resilient to heat stress in a warming climate. This could lead to more efficient, sustainable, and humane farming practices. It raises ethical questions, of course, about animal welfare and the industrialization of life, but the potential to feed the growing global population more securely is a powerful driver for this research. With all this incredible potential, we must also confront the ethical shadows. The idea of human reproductive cloning continues to haunt the public imagination. By 2030, will it be possible? Scientifically, perhaps. The technical barriers are falling away, but the ethical and societal barriers are, and should be, enormous. The vast majority of the scientific community and governments worldwide agree that cloning a human being is unsafe and morally unacceptable. The process has an incredibly high failure rate in animals, resulting in miscarriages, birth defects, and premature death. Applying such a risky procedure to humans would be grossly unethical. Beyond the safety issues, human cloning raises profound questions about identity, individuality, and what it means to be human. Would a clone be their own person, or forever live in the shadow of their genetic predecessor? The psychological and social implications are immense. So, while rogue scientists might attempt it, it's almost certain that by 2030, human reproductive cloning will remain firmly in the realm of forbidden science, regulated by international laws and treaties. The focus will remain squarely on the therapeutic applications that save lives, not those that try to replicate them. So, what does the world of 2030 look like through the lens of cloning? It's a world where a patient with heart failure might receive a patch of new heart muscle grown from their own skin. It's a world where conservationists might be preparing a habitat for the first mammoth hybrid calf in 10,000 years. It's a world where our food is more secure and our ability to fight disease is more personalized and powerful than ever before. The journey that began with a single sheep in a Scottish laboratory has led us to the brink of a new era. Cloning is no longer a distant sci-fi concept. It's a powerful tool that is reshaping medicine, conservation, and our relationship with the natural world itself. The future is complex, and the path forward is lined with both incredible promise and profound ethical responsibility. But one thing is certain: the world in 2030 will be a place that Dolly could only have dreamed of. Thank you for joining us on this look into the future. What part of cloning technology are you most excited or worried about? Let us know in the comments below. And if you enjoyed this journey, don't forget to like, subscribe, and hit the bell for more explorations into the world of tomorrow.